Good afternoon. Welcome to the Walls Lecture. And today it is a great pleasure to introduce Dr. Tom Maniatis to you, and we're all certainly looking forward to a wonderful talk. Tom received his BA and MS from the University of Colorado at Boulder, and then went on and got his PhD in molecular biology from Vanderbilt University. He went on then to do his postdoc at Harvard and at the Medical Research Council Laboratory of Molecular Biology in Cambridge, England. He's also held a research position at Cold Spring Harbor Lab and was on the faculty of California Institute of Technology. He's spent a good portion of his career, 30 years, at Harvard, and I think this has been a really good time for him there. And then in 2010, he moved to Columbia University Center, where he is now. Tom has been really renowned throughout his career, from his early days in looking at innate immune system signaling through his current ventures in looking at the brain, and has been well recognized for his work over the years. He is a member of the National Academy of Sciences and a member of the American Academy of Arts and Sciences, the American Academy of Microbiology, and a AAAS fellow. So the awards he has are many to expel on, but he's had numerous awards. In addition, he's co-founded a number of very important companies: Genetics Institute with Mark Potashny, Proscript Pharma, and Acceleron Pharma. I think the accomplishments, not only from his science, but the application of his science to medical products that are now being used today, really speak not only to the basic science that Tom has accomplished, but his ability to translate those into products that help many, many people. So it's with great pleasure that I introduce Tom, who will talk to us about the generation and function of single-cell diversity in the mammalian brain. Tom, thank you very much for coming.
Thank you, Kathy, for that very nice introduction. I'm going to talk about work today that, strangely enough, even though it seems new, really started almost 40 years ago with a GMS grant that funded our work on recombinant DNA, leading to the cloning of the globin genes, which in turn led to our work on thalassemia, which in turn led to splicing. And this is the end point of, thus far, of the splicing work. So I'm going to talk about this wonderful gene cluster here called the protocadherins. Protocadherins are cadherin-like, calcium-dependent cell surface proteins. And the story behind this is that we were 10 years ago looking for a system to study alternative splicing in the brain. And a paper by Yagi appeared which showed a series of cDNA clones that had identical 5' ends, but identical 3' ends, but different 5' ends. So we decided to, and they're expressed in the brain, so we decided to look at those. And when we cloned these, we found this. And it was remarkable. This was before the genome was sequenced, human or mouse. And we had to cobble this together, and it was actually Chung Wu, a postdoc in the lab, who did this. And it turned out to be really complicated because all these sequences are repeated, as you see here. And I'll explain later does. But the bottom line is that we and laboratories of Josh Sains, Josh Weiner, Yagi, my former postdoc Chang Wu, have over the last 10 years obtained evidence that this gene cluster functions as a single cell barcode to give individual neurons their identity. And when we first cloned this, it obviously looked very much like immunoglobulin genes. We did all sorts of work to see whether or not DNA rearrangements occurred, and they don't. And I'm going to tell you, they do generate diversity, but by a different mechanism. So this is really sort of the biological problem that underlies this work, and that is that, and this goes back to Roger Sperry many years ago, in that individual neurons, he proposed, have a unique identity, and this identity is required to distinguish one neuron from an otherwise identical neighboring neuron. And what has emerged over the last 5 or 6 years is that the unique identity of neurons is generated by the combinatorial expression of these individual isoforms of cell surface proteins through a stochastic alternative pre-mRNA splicing or alternate promoter choice. And I'll show you the 2 examples. And this is work primarily from Zabersky's lab, which is really a beautiful set of studies that were carried out over several years, in which this amazing gene was identified in Drosophila, which has all these alternative exons, 12 of these red, 48 of the blue, 33 green. And by a stochastic splicing, alternative splicing event, it generates messenger RNAs with 1 red, 1 green, and 1 yellow, and 1 blue from here. And that, it turns out, encodes a immunoglobulin-like homophilic binding domain at the cell surface, and therefore neurons that splice in this way generate a code. So this gives rise to 19,000 different alternative cell surface splice forms. Different neurons express different combinations of these splice forms. And as I'll show on the next slide, self-recognition triggers repulsion, so that only strong binding between proteins of the same splice form interact. So these are not adhesion proteins. These are signaling proteins that, once engaged through homophilic binding, lead to downstream signaling, which signals repulsion. So this just shows it in a more structural way, where if you have a perfect match, you see that these 3 immunoglobulin domains engage, and you see binding. And if there's a single difference in an isoform, you see no binding. They even went on with Eisenberg, to determine the structure of this homophilic dimer, as you see here, and really were able to map out in great detail the sequences required for this homophilic interaction. So, of course, in Drosophila, you can do extensive genetics, and that's what Zabersky's lab did. And he could show the following, that in wild-type neurons, the 2 neurons cross each other, but they do not cross themselves or interact with themselves. However, if you look at a DSCAM mutant, you see that it does cross others, but it also crosses itself. So it clumps, it forms these aggregates like this. And if you have a single DSCAM isoform, it recognizes others as self, and therefore repels. So the basic idea, although this has not been shown at a molecular level, is that when you have 2 neurites and they have identical homophilic interactions with neurite A and B, they engage a signaling receptor ligand that's disengaged through a mechanism that's not understood, and that, again, by a mechanism not understood, leads to repulsion. So the question became then is, what is the cell surface diversity in vertebrates? That is, what is the source? And when the first DSCAM genes were cloned and characterized in mammals, it turns out that they're very different. They do not have the extensive exon arrangement, and they can only generate a few distinct isoforms. And so these clustered protocadherin genes have merged as the best candidate for generating this cell surface diversity. So here's a larger view of the cluster, and I want to point out just a couple of things. First, it's a million base pairs of contiguous DNA sequence, and you'll see it's highly conserved among all vertebrates. And it consists of the following. There are variable first exon, these orange guys here for protocadherin alpha. And in the alpha and gamma clusters, there are these pink C1 and C2, which we call the C-type isoforms. They differ from the isoforms within the cluster, but they're very similar between clusters. So C1 through 5 are very similar. The beta cluster also has all these isoforms. It's expressed in the same manner as these, but you see it does not have a constant region, so it does not have an intracellular domain, as you'll see. The protocadherin gamma cluster looks like this. There are two types of protocadherin gammas, A and B, and again, C3 through 5. Now, each of these clusters are expressed independently, and you can see here from the extensive sequence of everything from man to coelacanth that if the general organization is highly conserved throughout vertebrate development. So we began, when we discovered that that the variability was not generated through a DNA rearrangement, we began to look very carefully at the mechanism. And what we discovered, which I'll elaborate more in a moment, is that each of these variable regions, which encode the extracellular domain, has a promoter immediately upstream. So what would happen in this random case here, the protocadherin alpha 8 promoter would be activated. It generates a long transcript. It looks like this. This transcript, if it initiates here, is as long as 300,000 nucleotides long. It transcribes to this entire region, ignoring all the 5' splice site in these exons, and only the cap proximal 5' splice site is chosen to splice to the first constant region exon, which is the first 3' splice site. This generates a messenger RNA containing the variable exon 8, now spliced to the constant regions, which you see is the intracellular domain. So this protocadherin alpha 8, these are cadherin-like EC domains. There are 6 of them, transmembrane domain, and the constant region, which, as you'll see, we think plays a very important function. So I'd like to begin by discussing the mechanisms for generating diversity in this system, both at the level of gene expression and at the level of homophilic interactions. So the function, and then I'll go on and at the end discuss the function of protocadherins in neural circuit assembly. So this diagram here, it makes a very important point. And it was work that was done in Yagi's lab in 2005. And basically what was done was to take 2 inbred mouse lines, cross them, take individual Purkinje cells from the F1 progeny, and do single cell sequencing. And this was the old-fashioned way by making cDNA libraries and sequencing. And what you can see is that what they ascribe here is so-called monoallelic, and you see this is kind of an odd use of the term. It's not really monoallelic, it's independent allelic, stochastic expression. And so what you see is that in the maternal allele, for example, in a typical case, alpha 7 will be activated, and in the paternal allele, alpha 2 and alpha 10. While the C genes both have promoters, and they're activated biallelically in every cell. So in this neuron, the monollelically expressed are protocadherin alpha 2, 7, and 10, and the biallelically expressed are protocadherin alpha C1 and C2. So if you look at this, then you see there are 14 alphas, 22 betas, 22 gammas, and it turns out that both the beta and gamma clusters are also expressed in the same way. That is, the 2 chromosomes are independent of each other. So if you tally that up, you can generate at least 3 million distinct combinatorial possibilities which is obviously far greater than that with the dsCAM proteins. So how does promoter choice work? So the first thing, if you look at the nucleotide sequences of the cluster, you discover that there's a highly conserved sequence immediately upstream from each of these variable exons. If you look closer, you can see that it has a consensus sequence, and that consensus sequence is present in all three clusters, in alpha, beta, and gamma, very highly conserved. So, of course, the first thing to do is to ask whether that consensus sequence is a promoter, and this was done by the now classic reporter assay of fusing the promoter to a luciferase gene. And what you can see here is that this is the full promoter, this is the level of luciferase activity. You delete down to the CSE, this conserved element, and it's active. Once you delete that element, it's no longer active. So in cells in culture, it does function as a promoter. Now, we then looked through this cluster for regulatory sequences, and we did that by looking at DNA's hypersensitive sites and using cells that do not express them as a control. And we found a number of sequences that were both highly conserved and DNase I hypersensitive. And we tested a number of them first in cell culture and then in mice. And the two that came out, as you'll see in a moment, these really are the only ones. This is hypersensitive site 7 and hypersensitive site 5 through 1. One's located in the intron here, one's located immediately downstream. So each of these elements were fused to LAC-Z reporter. And as you can see, in both cases, these enhancers drive expression of the LAC-Z in a nervous system specific manner, in a manner that's indistinguishable from the endogenous gene. So we deleted those enhancers and looked at the homozygous deletion mice and discovered that when you delete, this is the case of the HS51, you can see that it affects the level of expression of the entire cluster, although it seems to have a preference for certain promoters and certainly affects this sort of alpha 6 through alpha 12 in a much greater way in C1. You see that 5.1 does not affect the C2 promoter. So that enhancer clearly affects expression. The second enhancer also decreases. This is now homozygous HS7 delta mouse. And again, you can see the effect is not as large as was seen with the HS51, but you clearly see a decrease. So it appears that these two enhancers work together to achieve high levels of protocadherin. When I say high levels, I should say normal levels, because this is gene is expressed at a very low level. So the conclusions are that this 5.1 enhancer is required for maximal levels of expression of the alternate protocadherins, alpha 1 through 12, and alpha C1, but not C2. And there appears to be two expression domains, 1 through 5 and 6 through 12 in C2. The HS7 enhancer is required for maximal expression of all the protocadherin alphas, and there appears to be a complex interplay between the two enhancers and promoters. So the next thing we wanted to do was to identify proteins that specifically bind to the enhancers and promoters. And it was started off with some biochemistry and later led to in vivo experiments. And I'm going to show you some data that was taken from this SK SH neuroblastoma cell. And the reason is that obviously you can't do this in brain. If you're trying to correlate expression with binding, and every cell is expressing a different set, you can't do that. So we're fortunate to find a diploid neuroblastoma cell in which the protein DNA interactions can be studied in a single cell, a single cell population. So this is just the RT-PCR of this cell line. So it's called SKNSH. It's a human neuroblastoma cell line, diploid. It can be differentiated with retinoic acid. And what you see here is that it expresses alpha 4, alpha 8, alpha 12, and the two C2s. So this is exactly what Yagi saw for a single neuron, this kind of distribution. We don't know which of these are expressed on which chromosome. And what's interesting is that there was another line that was derived from this over 40 years ago, and both of these have been maintained in various labs and cultures. And you can see that they are identical. They express exactly the same set of protocadherins. And so once this choice is made, it appears that it's permanent and it can be propagated through cell division. So here's an experiment which we're doing, whole genome ChIP-seq, using antibodies, in this case, against the H3K4 trimethyl, a mark for active promoters and enhancers. And what you can see here is that these are just replicates. And you can see alpha 4, alpha 8, alpha 12, C1 and C2, and you can see marks at the enhancers as well. So there's a perfect correlation between active promoters, modified histones, and the expression pattern. Now, some in vitro experiments suggested, both by sequence and by biochemical experiments, that CTCF binds here. As you know, CTCF is an insulator protein that was made famous by Gary Felsenfeld, that binds to insulators and later was shown to engage enhancers, promoters, and be involved in a variety of positive and negative control of enhancer-promoter interactions. And what you can see here is that just as we saw the active genes correspond to the histone marks, we can see that both CTCF and cohesin, a subunit of cohesin, RAD21, binds to two sites, actually, in each of the active promoter, alpha 4, alpha 8, and alpha 12. So that general pattern emerged, and very deep and thorough studies revealed that, so this is the set of genes that are expressed. You can see they're all of these enhancers and promoters are positive for H3K4 trimethyl. All the active promoters bind CTCF. Of the alternate ones, they don't bind to the enhancer and the C2, which is interesting because they are different than the 5-1 C1. But what you'll see here, if you look closely, all of these alternate isoforms have two CTCF cohesin binding sites, which are shown here. And so if we look more closely, what we see is that if this is the CSE of protocadherin alpha 8, we see this is where the H3K4 trimethyl maps. It's in a region of about 650 base pairs, so enough room for about 3 nucleosomes. And then it's flanked by the binding of CTCF RAD21 to the CSE and to the CTCF motif. And if you look at this motif along the entire alpha cluster, you see it's highly conserved. So I think this is unusual. In most cases, CTCF cohesin have been shown to bind to enhancers and promoters. It's rare to see it. In fact, this may be the only case in which it binds within a specific conserved sequence within the exon. So if we look at the methylation in that region between the two CTCF binding site, what we find is that between the CTCF sites, there's very low levels of CpG methylation, but downstream it's high. And in the silent isoforms, you see high methylation in both. So there's a general correlation between high levels of CpG methylation and lack of activity. So all of the promoters that are off are methylated, and the ones that are on are unmethylated in this region. Now, if we look more closely downstream at the HS51 enhancer, what we see here, these are the level of conservation of these sequences. And you see we found a CTCF motif here and here about a kb apart. And you can see that in ChIP-seq experiments, CTCF and RAD21 also bind to these two sites in vivo. And you can see that the modified nucleosomes, like in the promoter, are positioned between these sites. Now, what was interesting is that when we used an antibody against NRSF rest, the repressor complex that shuts off neuron-specific genes in non-neuronal cells, we discovered that in glioblastoma in undifferentiated SK-NSH cells, that you see that the NRSF rest binds to a site that actually overlaps the CTCF cohesin site. And in fact, if you look in mice in which we have deleted the HS51 knockout and look at the expression of these genes, of the protocadherin alpha genes, in both astrocytes and kidneys, you can see there's a dramatic increase in the expression of these neuron-specific genes in these non-neuronal cell types. In this case, it's 35-fold. So what emerges then is that in non-neuronal cells, HS51 acts as a repressor, so it acts in some way as a long-range repressor. And in neural tissues, it acts as an enhancer, activating the appropriate sets of genes. In this case, it would be alpha 8, C1, and C2. And the HF7 is also active. So that led to the question of whether there's DNA looping between enhancers and promoters. And this work was carried out in collaboration with Chong Wu, the person who originally isolated this, who's now in Shanghai, and Kevin Monahan and Chong's lab collaborated to do 3C analysis, in this case using as an anchor a sequence in HS51. And you can see that there are peaks at both alpha C1 and C2, alpha 12, alpha 8, and alpha 4. So there are contacts between the 5-1 enhancer and promoter at exactly the promoters that are active and contain the CTCF cohesin binding sites. And this is the using the anchor of HS7, and you see a very similar pattern. So indeed, these enhancers do loop and engage each of the active promoters. This is an experiment just showing that if you use shRNA knockdown of CTCF or RAD21, that in each case you can see a significant decrease. Other experiments, both in mouse knockouts and in antisense experiments, show that CTCF is required for protocadherin gene expression. So what emerges then are two interesting thoughts that will lead to a lot more experiments. And the first is that if you look at the enhancer-promoter interaction, so what I've drawn here is the HS51 enhancer up here, the protocadherin alpha alternative isoform here. And so the idea is that there's a double clamp holding the loop together at this CSE upstream from the promoter, and then another here. And the spacing is about the same. It's 1,000 nucleotides here and 650 here. And so the idea is that in some way this may be a mechanism that stabilizes the interaction and could even, for example, stabilize that interaction through DNA replication as the replication fork passes through this region. Now, the interesting thing is that there are multiple interactions between 5-1, HS7, and these active promoters. So we'd like to propose that there may be an active transcription hub in which these interactions occur. It could simply be a focus for RNA polymerase, but it's also possible that this is a dynamic structure that, in which at any given time one enhancer will engage one promoter. So we're very interested in probing this in more depth. The difficulty here, of course, is that the fact that both chromosomes are expressed, and so it's difficult to determine which looping is occurring on which of the two chromosomes. So I'd like to briefly discuss the function of the protocadherin proteins and begin by discussing homophilic interactions. So the classic cadherins look like this. They're calcium-dependent domains. Their structure has been determined, and a great deal is known about the requirements for homophilic interaction. In 2010, Josh Weiner's lab published a very nice paper in which they carried out an aggregation assay I'll talk more about in a moment, in which, again, they demonstrated that there's homophilic binding for protocadherins, that the EC1 domain is essential but does not carry the specificity, whereas the EC2 and EC3 domains carry the specificity for the homophilic interaction. And they showed that the interactions are entirely homophilic. Now, at the same time, Steffi Schalm, a postdoc in my lab, did protein interactome studies, discovered that the RET receptor, a transmembrane tyrosine kinase, interacts directly with the protocadherins, and in fact confers upon protocadherins a GDNF alpha dependent tyrosine phosphorylation of the intracellular domain. And there are 3 other labs that have been looking at other interactions, and it's clear that there are both partners and kinases and phosphatases that associate. So it's got all the characteristics of a signaling complex. What also was shown by a number of labs, and we were able to show that the process requires endocytosis, in that there is cleavage by a metalloprotease at the cell surface, followed by a gamma secretase cleavage, the release of the intracellular domain, and we can see it in the nucleus. We don't know whether it's functioning in a manner that's similar to that of Notch. But it's, the main point here is that this is clearly a signaling complex. In this case, it appears to be activated by GDNF alpha, but there are undoubtedly many other ways in which it's activated. So this is the assay that the Weiner lab developed. They used K562 cells in which there's no endogenous expression of the protocadherins, and what's really important is that they can be cultured as suspension cells, because you can't do this aggregation assay for cells that sit down on the plate. So if you transfect the cells with a single protocadherin isoform, you see that, and you shake to allow for aggregates to form, there's either no aggregation or aggregation. You see these wonderful aggregates because in this case, the protein has been labeled with a red fluor. So what emerged from his study is the demonstration that, as I said before, EC1 is required for the interaction for transbinding. EC2 and EC3 contribute to the specificity and is essential for the trans interaction. And they speculated that 4 through 6 may be involved in cis interaction or recruitment of co-receptors. Doing some biophysical measurements, they proposed that the active form of the protocadherin is a tetramer, and that based on the fact that when you isolate these molecules from the cell surface, they're in various forms that they can form in different pairs, alpha, beta, gamma. So we were looking at this, and if you look at the evolution of these proteins, the sequence similarity, you can really see 3 classes: the beta, the gammas, the C types, which cluster, as I mentioned before, and the alpha. So we systematically tested these, and what you see here, here's the control. If we transfect the cells with alpha-4 cherry, we don't see any significant aggregation. Same thing is true of alpha-6 and alpha-7. C1 doesn't work. C2, you do see some aggregates. Here's beta, they work. Here's beta, all these betas work very nicely. Here are the gammas, they form aggregates, and the betas are here, although the C4 you see does not. So we're able to identify the protocadherins that interact, and we're able to make this following table. So 22 beta, all the ones we tested, 22, form aggregates. and we found that all of the ones indicated here, 7 of 19 we tested, they all test positive. The C's are variable, so alpha C1 doesn't, gamma C4 doesn't, but the other 3 do. But the most interesting thing is that if you look at the protocadherin alphas, the ones that are neuron specific and the ones that we think play a very important role do not engage in homophilic interaction. So we puzzled over that, and with an additional experiment made the following discovery. So it turns out, this is just a repeat of alpha 4, and this is deleting the EC1 domain, which would be necessary for its homophilic interaction if it worked. But what we found is that if we co-express the inactive protocadherin alpha with any active form, in this case the protocadherin gamma B6, and it has deleted its EC1, so it can't interact with itself, you see robust interaction. And that's a general property that we could rescue all of the protocadherin alphas that are unable to engage in homophilic interactions with ones that can, and in each case you can delete the essential domain of the interactor to show that it's entirely due to the interaction of the alpha 4. This just shows that you can rescue it with everything. So here we have co-expression with wild type alpha 4. Here is the C type that works, a beta, a gamma, and a gamma C type. So any one of these can work. And so that reveals even a further complexity and diversity of the interactions. So we conclude that every protocadherin isoform engages in a specific homophilic interaction, either alone or in combination with other isoforms. The ability of alpha to engage homophilic binding requires co-expression of this active isoform and works with beta or gamma. The EC1 through 3 domains are required for homophilic interactions in trans, while 4 through 6 appears to be required for cis interactions. And so what's really fascinating about this is that the protocadherin alpha, beta, and gamma isoforms provide a combinatorial code for this homophilic binding, and the code is expanded significantly by requiring cis interactions between homophilically active and inactive isoforms. So it increases the code substantially. And so this work in our lab is being done in collaboration with Larry Shapiro and Barry Honig, and we're very eager to look at this problem on a deep structural basis. So I want to finish with a very brief overview of functional studies which have been based on either the complete knockout or Cre-dependent deletion of one of or more of the gene clusters. So here's alpha, beta, and gamma. So the gamma was knocked out by Alex Wong's lab several years ago, and he collaborated with the Sainz lab to look at the phenotype. And what you see here is that the protocadherin deletion is neonatal lethal. It dies at P0. You see no coordinated movement, significant cell death in the spinal cord, and reduced synaptic density. Whereas if you now cross that mouse into a Bax homozygous knockout, it's still neonatal lethal. You see a little better movement. Of course, the apoptosis is blocked, and you see some reduction in synaptic density. So it wasn't terribly informative with respect to the function of these protocadherins. And so Wei-Xing Shen in my lab created a deletion that turned out to be very informative. So here's the wild-type protocadherin gamma gene cluster. Here is the entire deletion. And here is a deletion in which he just simply deleted protocadherin gamma C3, 4, and 5. And this was published in Neuron last year. And I don't want to go into detail with this, but what the bottom line is that the phenotypes of the gamma C, this one, and the full knockout, this one, are indistinguishable So the Cs are responsible for the cell death phenotype, and that provided a very important way of then looking at the function of these. It's important to point out that obviously you can imagine this deletion just removes functionally all of these because they're not transcribed, they're not spliced, they're not functional. But in fact they are by many different criteria. So here is the wild type. This is just looking at RT-PCR, the expression of all of these isoforms. And you can see they're all expressed in the C knockouts. They're obviously not expressed in the full deletion. We see expression at the protein level, and we can see them at the cell surface. Normal RNA levels, protein-protein interaction, and normal intracellular signaling. So they're functional, and the only thing they lack is this C region here. So we can conclude from that these C-type protocadherin gammas are required for neuronal survival, while the rest of the cluster is required, well, as you'll see, for circuit assembly or synaptic function. And in particular, the one solid evidence we have is with neuroid self-avoidance. So we are collaborating with Josh Sain's lab and with Julie Lefrave. And she had looked at both a full cluster knockout and was looking at our C knockouts. And she sees this pattern. So this is the wild type situation. This is with the homozygous knockout, and you see a whole loss of a whole layer of interneurons in this interplexular layer, and so you could see this, and as I said before, you see exactly the same thing with the C knockout. That's what's shown here. So both in the spinal cord and the retina, the full cluster deletion or the C knockout give exactly the same phenotype. So she could then look more carefully at amacrine cells within this layer. And as you know, these retinal starburst amacrine cells are layered like this. They're tiled in the interneuron layer, and they interact with each other horizontally like this and across this interplexiform layer. So they interact here, but they don't interact with themselves. However, and so this is a beautiful picture that shows how, in 3 dimensions, how they're layered in this way. And this is how it looks from an angle above. And so here is the wild type, and here's a cell-specific, cell-autonomous deletion of the protocadherin gamma cluster. And you can see that you completely lose this beautiful dendritic starburst configuration, and it looks very much like the Drosophila DS-cam mutants that clump and cross is shown in these 2 slides. So it's clear that they're required for self-avoidance, the protocadherin gammas. Here is the C knockout, and which was the important point here is that when you delete the Cs, as I told you, you express normal levels. And even though you see that cell death phenotype, you see that the starburst amacrine cells display normal dendritic self-avoidance. Interestingly enough, though, in exactly the same way as was shown for the Drosophila DS-cams, that if you rig it in such a way that you're expressing only protocadherin gamma C3 in a knockout background, you can at least partially rescue self-avoidance. And that was, as I say, also shown in DS-cam. So, the second area that we looked at was the protocadherin alpha genes. The protocadherin alpha deletions are viable. They grow to adulthood. They have no obvious phenotype, and they've been very difficult to study, but very recently are starting to yield interesting information. So I'm showing this as a paper from Yagi's lab, and most of the cells in the brain are expressing all three isoforms, alpha, beta, and gamma. But what you see in this staining experiment here, this is a protocadherin alpha stain, this is a protocadherin gamma. And you can see there's highly preferential expression of the protocadherin alpha genes in the dorsal raphe, in the serotonergic neurons. What they showed was that they were looking at not a full knockout, but a weakened mutant. And what they demonstrated was if this is the wild type, you see a more or less normal spread of serotonergic fibers through the brain, whereas in the mutant, there was a high concentration here. And if you can see this at higher magnification between the wild type and the protocadherin alpha mutant, and it's quantitated here. So we repeated that, and you can see exactly the way they show. This is a full knockout. You can see this amazing clumping or aggregation of the serotonergic neurons in this region of the brain, and they're clumped in the CA1 region. This is a close-up, and this is the wild type case where you see more or less uniform distribution of the serotonergic neurons, and it's highly clumped with the mutant, with the protocadherin alpha deletion. Interestingly, their fibers are depleted from the dentate gyrus. Again, you see here's the wild type, a more or less evenly distributed serotonergic fibers, whereas in the mutant, you can see that it's considerably less dense, sparse. Now, when you look over a time course, you can see that it does not change from the very earliest time we looked at, a week, all the way through 2 years. You maintain this stable distribution of serotonergic neurons. So the question that Wei-Xing asks is whether the serotonergic wiring is cell autonomous. And he used a SERT and a CAM-Kinase2 Cree in order to do that, one which is forebrain specific in the target field, one which is serotonergic neuron specific those derived from the dorsal raphe. So what you can see here, this is the two knockouts. This is the SERT knockout, so of course you don't see that. And what you can see here is that you see the phenotype when there is a cell-autonomous SERT-specific Cree, and you don't see that when you knock out the protocadherin alpha in the field. So this is clearly cell-autonomous, and there's no effect of deleting protocadherins from the rest of the brain. Now, he carried out a whole series of behavioral tests, and the most striking thing to emerge was changes in anxiety and depression from these open field, light dark box, marble bearing, fair conditioning, swim test, and tail suspension. They tested positive in each of these cases for the protocadherin knockout, and most importantly, the changes are really correlated. So here is the mice in which we did the serotonergic specific deletions. You can still see differences, the red and blue lines, in the tail suspension test, forced swim, in the contextual fear conditioning. But if we knock out the protocadherin alpha in the rest of the brain, you can see that there's no significant effect. So it's clearly cell autonomous, and the behavior correlates with the anatomical changes in the serotonergic fibers. So in summary, the C-type protocadherin gamma isoforms are specifically required for neuronal cell survival. The C-type isoform knockout uncouples the cell death phenotype from the neural circuitry phenotype. The protocadherin gammas are required for dendritic self-avoidance in the mammalian nervous system, functioning in a similar way to that of the fly DS-CAM isoforms. And finally, protocadherin alphas are required for normal serotonergic projections and effective function both roles of which are cell autonomous to serotonergic neurons and independent of the target field. And then what became even more interesting is that during the past year, a very large number of whole genome, whole exome trio studies have been carried out looking at unaffected parents and an affected child. And there are now many more studies since then. And basically, it was found that both clustered and non-clustered protocadherins are implicated in autism because you can, in each of these cases, among the 40 or 50 some genes that were implicated by looking at a very large number of children, the protocadherin alpha, beta, gamma, plus the unclustered protocadherins show up. So one can imagine that this is the kind of wiring defect in autistic children that can affect the behavior through, in this case, the projection of serotonergic neurons. So I'd like to thank the people in my lab that did all this work. Kevin Monahan, who's now a postdoc at UCSF, did all of the work on the protein-DNA interactions. Polina Kayahova did the studies on the enhancers. Etu Chan, a postdoc, did all the work on the homophilic interactions. Weixing Shen, a postdoc, did the work on the knockouts, and he was aided by Chiamaka Nakawazi and Maxime Chavy, technicians. We are collaborating, as I said before, with the Shapiro and Honig labs to look at the structure and the basis for homophilic interactions. We had a very productive and fun collaboration with Josh Same's lab on the self-avoidance. Rick Myers helped us tremendously in the ChIP-seq experiments and the sequencing, and the looping experiments were carried out in collaboration with Chang Wu's lab in Shanghai. Thank you.
So I was just wondering, I may have missed it. Did you say in the non-neuronal cell types where you have NSRF rest bound to the HS51 enhancer, that CTCF is also bound, co-bound, or it's not there?
No, it's not bound. It was just to indicate that they bind to the common site. And actually, it's quite interesting because it turns out that the beta and gamma clusters are expressed in many cell types, including the kidney. And there were some very interesting papers that came out a few years ago in which they're looking at Wilms tumor, looking at DNA methylation through the protocadherin locus. And they discovered that in normal kidney, the alpha is highly methylated and off, the beta and gamma are on. But in Wilms tumor, and now several other tumor types, the entire cluster is methylated and there's no expression. And they made some weak arguments that gamma C3 is actually a tumor suppressor gene. So these non-alphas are playing an important role in other cell types, and very little is known about that.
And one other quick question is whether CTCF is involved in regulating splicing at this locus.
You're saying, asking if it is?
Yeah, if there's any evidence.
No, it's really an interesting and important question because we have no idea why it is that 5' splice site at the end of the cluster is not reactive. I mean, that all the rest of the sites are not reactive and only the 5' splice site. So there could be a relationship, as I guess you're implying, that the ones with CTCF cohesin in some way assemble a coupled splicing complex which allows that to happen. But a new postdoc is coming that's very interested in that problem, but we don't know.
So it is my understanding that certain individual neurons can synapse onto themselves. And so I wondered whether this homophilic dendritic avoidance is specific to dendrites or or axon to dendrites, or whether we have to think of this code to be expressed in a cell domain specific way.
Yeah. No, that's really an important question, and you know, when you stain cells, that you can see the protocadherins at the synapse. Obviously, they don't match, and so what they're doing there, we have no idea. And there certainly is no evidence yet that they function in any positive way at the synapse, and that's a whole question that I think the field is struggling with now, is to try to understand whether they have additional functions than self-avoidance and actually participate in synaptic interactions in some way.
But you have no evidence for any code of combinations of protocadherins to be expressed within the same cells but in different domains.
Yeah, no, that's a really, and actually we are working very hard on that. We're collaborating with a group on single molecule detection by in situ and by fluorescence, and we're really hoping to be able to answer that question, because that's an obvious thing, that if they're assembling randomly, that perhaps they're assembled in the soma in a different way than they are in the dendrite or in the axon and so on. So it leaves open a huge number of interesting possibilities.
So I have a cancer-related question. So I was pretty intrigued by the neuroblastoma cell line that expressing a sort of a clonally defined protocadherin locus. So do you think this contributes to sort of the repulsion of a clonally arised neuroblastoma cancer, that sort of contributes to the invasiveness and migration of these cells?
Absolutely, it really could. Yeah, and I think that, you know, as I say, it's been implicated in other cancers, and there may, controlling the level of expression may in some way contribute. But we have no data to address that.
Have you asked a question if these genes, deletion of them would have a phenotype if deleted postnatally?
Yeah, no, that's a really good question. We obviously have the reagents to do that. It's a matter of doing the crosses, but it really is important to determine whether they require persistent expression or whether they're a developmental circuitry.
And possibly in certain, maybe in dementia, they could be involved in some of those events.
Yeah, no, and it would be interesting. For example, these are expressed in the rapidly dividing stem cells in the kidney. What happens if you knock them out there? Does it affect how the kidney tubules are assembled? And actually, Frank Cosentini, who is at Columbia, is very interested in this problem and has the mice to look at that carefully.
Do you have any way to detect endogenous isoforms and see where they're localized in particular neurons? And related to that, could you put a MIG tag, an HA tag in one of the loci, knock it back in and see where that goes?
Yeah. No, that's a good question. It was kind of the same questions before. It's turned out that it's been extremely difficult to get isoform-specific antibodies for the cluster. I don't think anybody has them. And even antibodies against the constant region are not very effective. So many people have wasted a huge amount of time trying to get that to work. So we don't know. We do have tagged forms, and we're working on that now. But we don't have any evidence that there is selective localization.
Thank you very much. I once again want to thank Tom for a wonderful talk. Please join us in the library. FAES is sponsoring refreshments, and you'll get a chance to talk to Tom if you get a chance. Thank you.